Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning platform in Immunology. Today we learn about general properties of antigenicity. The ability of the antigen to produce and react with the products is known as antigenicity. So it is the behavior of the antigen to elicit the immune reaction and to cross-link the products of the immune reaction. So for that molecule to consider it as the antigen, so what all properties it should serve. So this is regarding the general properties of antigenicity. It depends on the foreignness, molecular size, chemical nature and solubility. First we'll take up the foreignness. The first and the primary requirement for any molecule to qualify as an immunogen are that the substance is genetically foreign to the host. It should be the foreign substance, or it is not the self molecule. It should be the non-self molecule, and it should be genetically foreign. It should be genetically foreign to the host organism in which it is establishing the infection. In nature, an immune response will occur to a component that is not normally present in the body. So inside the body, our body itself will show the reaction to the non-self molecule. It will not elicit the immune reaction to the self molecules or the body own cells. And these molecules which are present in the inside the host organism will be exposed to the immune cell during their maturation. So that's why usually the immune system will not raise the reaction against the own cells or the own molecule. Antigens from the related species are less antigenic. Antigens from the related species, closely related species, are less antigenic than that of the unrelated species, or the antigens from the members of the same species are less immunogenic than the other species molecules or the antigen. The more distant or the foreign the antigen source, the better the antigenicity. So that means if in evolutionary tree, if you find the distantly related organism, when the molecule will be injected, so they'll show the antigenicity compared to the closely related molecule or the closely related organism. Next, coming to the molecular size. For a substance to consider it as antigen, it must be of minimum molecular weight. To consider it as effective antigen, they should have the molecular weight more than 10,000 Dalton. So it should have 10,000, the molecular weight more than 10,000 Dalton. The best antigen tend to usually have the molecular weight approximately 100,000 Dalton. So this is not the general assumption, but it is usually to consider it a substance as the antigen, it should have the minimum molecular weight is 10,000. But there are certain exceptional cases also. For example, the histone molecules are poor antigens. Histone molecules are poor antigens and they have the molecular weight of 6,000 Dalton. Hemocyanin, which is having the 60,000 Dalton molecular weight, it is a very good excellent antigen. And the protein like insulin, it is having the 5,700 Dalton, is also very good antigen. So like that, the antigenicity, they play an important role, wherein the molecular weight also play an important role in considering the molecule as the antigen molecule. Next, coming to the chemical nature. Most of the naturally occurring antigens are proteins and polysaccharides, and these antigens are found to be comparatively more antigenic than the lipids and the nucleic acid. The proteinaceous substances are more antigenic, and then comes the carbohydrate. So proteinaceous and the carbohydrate, the antigens are more antigenic compared to lipid and the nucleic acid. Proteins are better antigenic compared to the polysaccharides of the same molecular weight. The synthetic polymers, though macromolecular in their size, they are not antigenic, while naturally occurring macromolecules such as proteins may be highly antigenic because of their higher internal molecular complexity, so that will give the more antigenicity towards the molecule. So this is lacking in case of the synthetic polymer. So synthetic polymer, they are less complex, molecular complexity is less compared to the naturally occurring the macromolecule. So that is regarding the chemical nature. If it is more complex, the antigenicity is more; less complex, antigenicity is less. Next comes the solubility. How soluble the antigen is. The non-antigenic of the synthetic polymer is also attributed to their insolubility in the body fluid. If the antigen is insoluble in the body, that will not be readily recognized by the immune system, and they will be less antigenic. But the substances which is not converted to soluble forms by the tissue or the enzymes are also not antigenic. If the antigen is readily soluble and it is readily recognized by the immune system, it is more antigenic compared to the insoluble substance which is not recognized by the immune cell. So that is regarding the solubility of the antigen. So first and foremost, the foreignness, it should be distantly related. It is from the distantly related organism. The molecular size should be more. If it is more than 10,000 or the 100,000 Dalton, it is more antigenic. Coming to the chemical nature, it should be more complex. If it is not complex, then it is less antigenic. It should be soluble and should be processed in the body to recognize as the foreign substance, or they consider it as the strong antigen. Next, we learnt about the blood group antigens, and there we studied about the isoantigens. Now, the same scientist, Karl Landsteiner, introduced the term haptens. The concept of haptens emerged from the work of Karl Landsteiner. Hapten, as it is a substance that can combine with an antibody but cannot initiate, see here, but cannot initiate the immune response. They are not immunogenic. They cannot activate the immune reaction, but they can combine the products of the B cell products or the T cell products, or they can combine the cellular immunity and the humoral immunity product, but they fail to activate the immune reaction by itself. So these are the substances. Haptens are the substances. They are not immunogenic, but they are antigenic, unless it is bound to a carrier before introduction or introducing into the host body. Before introducing into the host body, if it is combined with the carrier substance, then it could be a very strong immunogen. Hapten is a substance which cannot initiate the immune reaction but can cross-link the products of the immune response. So the haptens, when they are complexed with the carrier substance, they can activate the immune reaction, and then they have the inherent capacity of cross-linking the product of the immune reaction. Haptens become immunogenic on combining with the larger carrier molecules. For example, dinitrophenol may combine with the serum albumin. Dinitrophenol may combine with the serum albumin to form the complete antigen, so that it induces immune response. Similarly, some substances act as haptens in some animals, whereas they act as complete antigens in other organisms. So the haptens, you cannot generally say this is the molecule which acts as antigen in all the species. So in some organism they may act as hapten, but in some other organism belonging to the different species, they may act as the complete antigen, where they are the eliciting the immune reaction and also cross-link the products of the immune reaction. For example, pneumococcal polysaccharide may be a complete antigen in the animals, such as the mouse and man, but it acts as the hapten in the rabbits. So you cannot generally say it is the hapten molecule in all the organism. Many drugs and peptide hormones and steroid hormones can act as haptens. So haptens are not immunogenic, but they are antigenic. But when they are complexed with a carrier molecule, they are very strong immunogens as well as the antigens. They are the complete antigens. So this is very very important as far as your short notes is considered. There it will be asked for both 2 marks as well as 5 marks question. Next we will move on to the term epitope or antigenic determinant. Epitope is the portion of the immunogen or the antigen that binds specifically with the membrane receptors on T lymphocytes or B lymphocyte. So it's a portion of the foreign substance that interact with the receptors on the T and B cell. It is called the epitope, or it is otherwise named as an antigenic determinant. So on the surface of the foreign substance, we'll find the certain molecules, and these molecules specifically interact with the B cell receptor or the T cell receptors. So they are called as the antigenic determinants or the epitopes. So these epitopes can be defined as discrete sites or the specific macromolecules which is recognized by and binds to a lymphocyte receptor. So they can also be defined as, these are the discrete sites on the antigen, on the surface of the antigen. They will be specifically recognized by the lymphocytes. An antigen may have more than one such epitope. So one antigen can have more than one epitope. So the number of the epitopes present on the given foreign substance or the antigen is referred to as its valency. For a protein, an antibody binds to about 3 to 6 amino acid for about 5 to 6 sugar residue on the antigen, which is made up of the carbohydrate. That means if the antigen is proteinaceous, about 3 to 6 amino acids on the foreign substance can interact with the antibody. If the foreign substance is made up of carbohydrate, if it is made up of the sugar, 5 to 6 sugar moieties will be interacting with the antibody. That means the epitope length may be around 3 to 6 amino acids or 5 to 6 sugar residue. Therefore, most large molecules possesses many antigenic determinants per molecule. That is, they are multi-determinant. As we said, it is the valency of the pathogen. So since the epitope size may be around 3 to 6 amino acid or 5 to 6 sugar residue, the one foreign substance or the one pathogen can have more than one epitopic region or the antigenic determinant. However, these determinants may be identical or different from each other on the same molecule. The determinants may be identical or different. We'll find the in case of protein, we'll find the different the configuration, like the primary configuration, secondary configuration, tertiary and the quaternary configuration. When the folding is different, we will find the arrangement of the molecules or the arrangement of the amino acid in the sequence of the arrangement of the amino acid will be varying, due to which you may find the different type of the antigenic determinants or the epitopes. So such type of epitopic region may be identical or different. For example, in case of carbohydrates with repeating sugar units, we'll have these several identical determinants. Well, the large single chain protein will have units each with different amino acid sequence. So thus the different antigenic determinant will be observed in case of the protein. But in case of the carbohydrate, if they are made up of the repeating unit of the same monosaccharide, you'll find the identical type of the epitopes. But in case of the proteins, since they show the different configuration, the arrangement of the amino acids on the surface of the epitopes will be differing, and hence you'll find the different types of the antigenic determinant. So these epitopes or the antigenic determinants also very important as far as your the examination is concerned. So and the foreign substance may be having the identical antigenic determinants or the multivalent antigenic determinant, and the different type of the antigenic determinant are usually more in case of the proteins compared to the carbohydrates. Okay, so in particular pathogen, we will find the different antigenic determinant. So in this session we learned the general properties of the antigenicity, and we learned about the haptens and the epitopic region or the antigenic determinants. I hope we understood this topic in this session. Thank you.